Can you all hear me all right? Because do excuse me if I'm coughing quite a bit today. Well, my name is Ryan, and here I am presenting my capstone study on the effects of biochar seed encapsulation in mitigating salinity stress and allelopathy. So first, let's take a look at what is biochar. Well, biochar is a form of organic material that's produced through a heat-treated process or pyrolysis. It typically carries a negative surface charge as well as having a very porous surface makeup. So using biochar as a form of soil remediation has accumulated a good amount of research attention in the past decade because existing literature has found out a 40% increase in crop yield when we integrate biochar into soil remediation. And also being a very sort of porous structure, biochar is a great growth inhibitor absorbent. So it absorbs heavy metals, salt, and phenolics in the soil. And also being a woody byproduct, biochar has the inherent advantage of carbon sequestration, and it is projected to offset 15% of the global agricultural carbon footprint if biochar is fully implemented. So all these sort of benefits that I talked about so far is sort of utilizing biochar in a traditional manner, almost using it as a form of fertilizer. But then relatively less research is dedicated towards biochar seed coating. So why are we doing seed coating? Well, seed coating offers a couple of benefits. First, it's a form of protection. So it has its logistical advantage when you transport the seed for longer distances. And also the encapsulated seed can be integrated into a high throughput seeding approach, such as this one right here using a UAV. And also, if you remember that picture I've been putting up there for this many minutes, the encapsulated seed is not that different from unencapsulated commercially available seed. So when it comes to your end user experience and how you deploy them and how you integrate into your day-to-day agricultural practices, it's pretty much the same and it's pretty intuitive. But then when it comes, I have this thing. But then when it comes to biochar, and especially seed coating, it's not just all rainbow and sunshine, right? Like we're still not sure on what binder we can use. And so far, the most common binder that we have is PVA or polyvinyl acetate. And this is PVA in its sort of natural form. So would Elmer's glue be good for my seed? And also we're not sure on germinant's physiological response. So how would the seed physically respond after being coated by this white and thick material? And also finally, would biochar seed encapsulation also be an effective sort of soil remediation method and would also lead to that 40% crop yield? We're not sure on that either. So for this project, I looked into using biochar as my seed coat and then using PVA as my seed binder. And I looked into these three questions. Well, first it's kind of covered up, but will the seed coating enhance germinant's development and crop yield? And will allelochemicals and salinity suppress germinant's growth and mature plants development? And finally, will biochar seed coating sort of mitigate the negative effects that I introduced with salinity and allelochemicals? So to look at my experiment, first I have two types of seeds. So the one coated with biochar and the one that's uncoated with biochar. Then I have 5 different environments. I have 2 different salinity treatment, 2 different allelochemical treatment, and then my distilled water as a control. So to carry out my germinations, I have 5 seeds from either coated or uncoated, put into one of each environment, and each environment is represented by 1 petri dish trial, and I have 3 replicates of that same environment for statistical significance. Then all the germination was then happening in a germination chamber that's happening indoor at 24 degrees Celsius room temperature with the growing light at on top that simulates a 12-hour day to night schedule. So that thing was let them to develop into little germinants. And then after day 4, day 8, and day 12, after the onset of germination, morphological parameters were taken. And then on day 15, I transplanted the germinants into a greenhouse to let them develop into mature plants. And then on day 5, day 10, and day 15, after my transplantation, physiological measurements were taken. And then on day 20, that was the day I harvested the radishes, and that's the day I measured the yield. So I'm sure you guys are wondering, what are these parameters I'm talking about? Well, for yield, it's pretty intuitive. It's just my root mass, so how big the radishes are. This picture doesn't show up that nicely, but these are nice and pink radishes. Then my morphology, that's my percent germination, so the amount of seeds that germinated, and also the radical length, so the root hair that's developed in my germination stage, how long those are. And then finally, for my physiology, I used leaf chlorophyll contents as a physiological indicator. And to measure those, I used a chlorophyll fluorometer to give me a accurate, relatively accurate reading when I'm trying to measure leaf chlorophyll. So to look at some of my results, first with salinity, I see a pretty big reductive effects when it comes to non-biochar coated seeds. So looking at these two right here in comparison with the blue bar in the blue bar in the middle, I see around 50% of reduction when it comes to germination rate. And also I see comparative level of reduction when it comes to my allelopathic treatment on the non-biochar coated seeds. Then looking at seed encapsulation, so that's the gray bars right next to the blue bars, I see my germination rate is actually lower in all my treatment and controls. And in fact, my biggest reduction is happening in my distilled water control with 82% of reduction being observed. And then, looking further, the other morphological parameter, which is my radical length, I see pretty much similar level of reduction. I see 75% reduction when compared with 40 millimole of sodium chloride with my distilled water. Then I see 53% reduction when I compare with the lower sodium chloride concentration with my distilled water one. And again, with my allelopathic, I see, pretty big reductive effects on that end as well. Then with seed encapsulation, I see it went down in all my treatments and control in comparison with the biochar coated one, with the non-biochar coated ones. So sorry, the blue bars here. So all of them are reducing beside my 40 millimole of sodium chloride one. So what's happening in there? And then looking at my root mass, so that's my yield measurement, I see a 55% reduction in my 40 millimole of sodium chloride treatment in comparison with the distilled water one. So this blue bar versus this blue bar in the middle, I see a 55% reduction. And then with my allelopathic treatment and my other, lower concentration sodium chloride treatment, I didn't see a very strong signal. And then with seed encapsulation, I think this is when things get interesting, is that I see a huge jump in terms of root mass in all my treatment and control. And in fact, even my distilled water control, this thing went up by around 50%. And the one that's doing the worst before, which is my 40 millimole of sodium chloride treatment, this thing jumped up by 65% as well. And then finally, it's all covered up. Finally, looking at my leaf chlorophyll content, that's what it's saying over there. My salinity once again reduced them quite significantly by around 40% in my 20 millimole sodium chloride and 40 millimole sodium chloride. Then my allelopathic treatment didn't have a strong signal. Then with seed encapsulation, I see a chlorophyll content increase in both of my salinity treatment, but it doesn't have a strong significant effect in my distilled water control and my allelopathic treatment. So just to recap some of those findings that we just went through, first, salt is, bad for my plants. That's, I guess, what I can conclude. It decreased my yield. It made it perform worse, both morphologically and physiologically. And then my allelopathic treatment, it did reduce my germination percentage and my radical length, but it barely did anything in terms of yield and my physiological response. And then looking at biochar seed coating, it did increase my yield, so my root mass and my chlorophyll contents, but it significantly reduced my morphological output, which is my germination percentage and radical length. So at this point, I'm wondering, so first of all, what's the mechanism behind why salinity is bad? And then also why allelopathic treatment that I introduced in this scenario didn't do what is intended to do and stress out the plants. And then finally, and I think more interestingly, is why my biochar seed coating, which I introduced them to sort of promote early germinants growth, actually decreased both my germination rate and radical length. So to answer these questions, first I looked into the structure of my seed coating and if it's going to have an inhibitory effect. So looking at this is PVA or polyvinyl acetate, and this is the binder that I use. And without getting into too much organic chemistry here, we can see this is an asymmetric looking structure. We see a huge functional group right here at the bottom, but there's nothing on the other side. So this is a nonpolar structure, and being nonpolar, this is a hydrophobic structure or hydrophobic substance. And what this means is that water cannot dissolve it. So essentially what happened is with PVA coating, there's a thing, film of unpenetratable layer that's being formed around the seed, and a prolonged exposure of that can actually decrease the equilibrium moisture content or EMC within the seed. And what this does is that because seed relies heavily on stuff like EMC to sort of signal its development and other metabolic process. So a prolonged exposure of that and decreasing EMC sort of reduced my water and oxygen intake and slows down other metabolic process, including germination. And sort of this is why it also makes sense when I transplanted my plants into a greenhouse, because the soil retention and the moisture content within the soil is a lot better than the what I have in the petri dish. So to sort of verify what I just said, I did another side project just on top of this. So this is just coating seed with PVA only, and then having another bunch of seed that's not coated. And I see a big reductive effects in terms of germination. Yeah, so after all that, let's look into why salt is bad. Well, typically we believe these two mechanisms. First is cytotoxicity. People believe that, a high concentration of sodium chloride is toxic to the cell and can cause cell death. So that's probably what happened. And also it's high salt is a form of osmotic disturbance. So what this is saying is that it disturbs the liquid balance within the cell. And being a cell, they relies on this sort of liquid balance to signal its development and next step in what a cell can do in their life. In both of these cases, both of these cases are a form of stresses. And when plants are stressed, they use an enzyme called chlorophyllase to start degrading its chlorophyll, because chlorophyll, if you think about it, is a very energy-intensive protein to make. So by degrading those chlorophyll, plants typically goes into this protective stage and conserve its energy and protect more of its vital organs and whatnot. And then looking at biochar seed encapsulation and why is it good? Well, because as we said earlier, biochar is very porous. So it increased the soil, nitrogen and phosphorus retention and other, nutrients retention in the soil. So it means there's more nutrients for the seed to uptake, which lead to a higher structural protein formation, which is my root mass as I measured, as I observed. And then being physically more massive, bigger, the radish will have a higher photosynthetic rate, which is what I measured which corresponds to my chlorophyll measurement as well. And then finally, I just want to touch on why my allelopathic treatment didn't have the effects as I expected. So looking at the treatment production process, it's pretty easy to tell that it's very prone to errors. The thing is collected on a field, stored on a field condition, and then hand refined. What I mean by that is me picking out the impurities and the gravels and whatnot. So this whole process is prone to errors. So to sort of improve this methodology, what we can do next time is to use a higher concentration, especially a weight to volume concentration, to sort of shotgun for more of those, important allelochemicals. And also another good aspect is just to use fresh leaves to entirely disregard the impurities of me picking litter from the ground can introduce. Yeah, and finally, I just want to sort of end on this future direction, because it's pretty obvious that PVA is not the optimum binder. It did reduce my early germinance growth. So what is? Well, I collabed with another lab at U of T, and we used Biochar as a different binder. So this is a hydrophilic binder, which is water soluble. And with some just very empirical findings, I can already see this did give me a much higher germination rate. So I feel like more time and resources should be devoted into this area to sort of push this study a step forward. Yeah, so that's about my seminar. I'd like to thank everyone who supported me so far, as well as the generous support from Seed the North. And I'm open to questions now. Very glad I didn't cough once, so this is great. Yeah.
Thanks, Ryan.
Yeah, thank you.
Natasha, you're with us virtually. If you could ask Ryan the first question.
Hello, thank you for your presentation.
Thank you. Thank you.
I'm interested to know how the very porous characteristics of biochar from your perspective could benefit from more greenhouse or fields studies, regardless of binder, or like a more mixed binder alternative. And the reason I'm asking this is because this seed encapsulation is from Seed the North's perspective intended for field application. And so we're similar to your conclusion that a biocode seed coating or an alternate seed coating is hydrophilic. And so I want your analysis of the porosity of biochar to reflect on the future direction in more greenhouse and field experiments.
For sure, yeah. Thanks, Natasha. That's a great question, because for the time being of this little presentation, I cannot include everything I have in the project I did. So in my actual project, I actually have an independent greenhouse trial that is happening. So I did include them in my appendix here. Yeah, so this is my greenhouse trial. So I have biochar seed, no biochar seed. Then I put them in a sort of soil salinity treatment and my watering salinity treatment. And with this, I actually found out pretty comparative result in comparison with what I did with the petri dish germination trials. So I see all my root mass went down in my in my non-biochar coated seeds when I salt stress them, and then my root mass went up when I coat them with biochar. So I think this can be a sort of direction now it goes into if we are thinking about more field application sort of approaches. And also another sort of aspect that we kind of talked about, and I sort of touched on this in my, in one of the earlier slides in my presentation, is that sort of utilizing biochar as a traditional form of soil remediation. So you mix them in with my potting mix, and that sort of methodology should also be considered, because I feel like, especially with doing this research, I feel like there's so many things that's happening, and there's so many different angles we can look at this. But then looking at efficacy and the effectiveness of remediation, I feel like that's the predominant driver that a company like Seed the North should be concerned about, I feel like. Back to you.
Thank you.
Yep.
Thank you. Any other questions from you, Natasha? No. Okay.
Okay, if you want to talk about the effectiveness of remediation, what kind of soils would you target as biochar being an optimal solution?
This is such an interesting question, because I feel like it really depends. It's kind of like asking, because the whole idea of me working with biochar up till this point is that it's a new emerging technology that we don't have much standards for it yet, such as what is your feedstock and where is this biochar getting derived? Is it from sewage material, or is it from coniferous species? And depends on what's the feedstock. The application approaches is very different. Just using an analogy that all of us in here can sort of relate, using mulch, right? Like your coniferous mulch can be used with certain different things, but not fruit trees, because it will acidify the soil. That sort of idea, I think, should be the area that we sort of examine and resolve before we can move further in this direction. Natasha.
Great, thank you. Sean, do you have any questions?
Yeah, I'll just ask a quick question here. So there's a bunch of kind of drone-based seeding companies working on this, and they've used a variety of kind of recipes to try to. You really have to coat the seeds with something so that your little mechanism to drop the seeds will work. And various things have been tried. So I've seen animal manure used. I've seen lake sediments used, kind of whatever they can fit in there. So I guess a question comes up: what is the fundamental motivation for having this sort of more technologically kind of based standardized coding mechanism for these? Why not use sort of whatever organic substance, sticky substance you can find to disperse your seeds in?
Oh, of course, that's a great question, Sean. So looking at where should I begin this? So actually, Sally asked me a question yesterday about why her lab hasn't heard anything back from Seed the North yet in terms of biocode seed coding. Well, I think I looked into a bit in that, Sally. They didn't have much success with seed coding. So and then like I talked to Esther, which is another member of the Thomas lab that's been working closely with Seed the North and this sort of coding technology. And the mechanism we use is extremely different. So for people who without a concept, there's typically two sort of methodology that you use to coat the seed with biochar. There's like the most sort of commonly accepted way is you dip them in the binder, and then you put them into this rotational device, and then the rotational device will have biochar in them, and then you sort of spin them around, and then the sticky part will get encoded by biochar. But for what I did in this experiment, and that's why I had this germination success in here, is that it's an entirely different sort of methodology. This is what I use. I'm sort of using, instead of like a spinning, sort of mechanism, I use like a pressing, force to make these sort of encapsulation. So going back to your question, Sean, I think the reason why we need these sort of standards is to standardize these, because when I'm doing these sort of seed encapsulations, there's so many different ways we can do it. I even bought a takoyaki press, and that thing didn't really work because the thing was too big. But that's sort of the idea that why standardization is pretty important, because to push this thing one step further, and then to make the market acceptance, and to achieve that 15% of global agricultural carbon footprint offset, I think that's the sort of the unavoidable area that we have to sort of tackle.
Great. Any other questions from the audience here? What about online?
Oh, Kevin has a question.
Yeah, one question. Go ahead. We've got a few minutes.
Yep. Yep.
So how long of an effect do you think it has on longer growing, like 20 days, did it have an effect on that? Like thinking about trees or thinking about corn or something that can grow all season. How long do you think that can go on? The biochar effect.
For sure, this is such a good question, Kevin. And this is the area that actually Esther is looking at right now, is that residuality of biochar seed coating. If you coat it with biochar, put them in the soil, how long would the thing degrade, and how long would this aspect sort of kicks in? That's one of the reasons why I use radish, because I kind of want to tackle that question, because I don't want to introduce all those confounding variables by having different species who has different development stage. So I use radish, which grows like crazy in 3 or 4 days. And to answer your question, although I cannot give you an explicit answer for that, but to answer your question, under the 4 days of my radish development, they were okay. But then I cannot give you a very solidified answer if I'm looking at, let's say, a hard, like a coniferous tree seed or what have you, because it's just not enough research has been looked into in that area. But that's why, there's dedicated master students from the Thomas lab that's looking into that area, and I'm excited to see what she pumps out at the end of the year.
That's great. I actually have a
There
question.
you go.
So the alternative to PVC coating
PVA, yeah.
that you, oh, sorry,
Yeah,
yeah.
that's
PVA
close.
Coding that you showed was like trademarked, it looked like. So where's that being developed and what is that? Like, what's it made of?
Sally, can I say this or? Oh,
Is it top secret?
it's
It's
not top-
fine if I've
it's not
touched.
top secret. Yeah, no. So this is a code that so Sally's lab I've been working with. I don't know how. First of all, I don't know too much detail about this, and also I don't know how much detail I can go into this. The primary application of biocode so far is actually not for this whatsoever. It's to make the that sort of liner within paper straws and whatnot to make it waterproof and also biodegradable, because there's so many people are saying water straw is kind of this ridiculous concept that, you know, it takes time to break down the water. It takes time to break down the paper as well, and how that sort of offsets our biomass and all that, you know, political debate behind it. So the whole idea of biocode is to sort of make your plastic substitute more biodegradable. So I don't know if that answers your question, Catherine, but
Interesting.
yeah. So
thank
this
you.
is the origin behind biocode C coding. And Natasha, I don't know if you're still listening. So I know you guys had a couple issues with biocode C coding, and I was trying to talk to Matthew about it. There's so many things happening with this material that I don't even know. But if you next time, if you're doing trials and whatnot, if you let it cure for 2 or 3 minutes before you start coding them with biochar, you might have higher success. And then also that press up, this prototype that I showed you, this might also be a thing that we can look into. But again, like I feel like your approach is a lot more, you know, industrializable than this, because this is a pain in the ass to make. But yeah, this is a good area we should look into.
Yeah.
Great. Thanks, Ryan.
All right.
Great job.